Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last class we started our discussion on dimensionality reduction techniques, and we talked about two different dimensionality reduction techniques. One is the principal component analysis and the other one is UMAP. So, in this class we will be talking about the principal component analysis. As you have seen, right, we started working on a data set. In the last class we have loaded the data, and in this class we will actually do the principal component analysis hands on. Okay, so this is what we are going to do in this class. We will actually demonstrate how we can do principal component analysis in R using different tools. One of them is PRComp function. This is a built-in function in R. The other one we will use is the Factomine R package. Okay, so as I said, principal component analysis is a linear dimensionality reduction technique, and we can extract the key features using the principal components, and these are the weighted combinations of original variables and what we refer to as PC loadings. Okay, so we will use this term and we will actually look into the PC loadings when we actually go into the hands on. For the hands on part, we will do the PC analysis on a cancer data set. So this is the data set that I just introduced in the last class, and we will work on that, and then subsequently we will be working on PC analysis of an yeast gene expression noise data set. Again that will be using a different tool, PCA tool. So in this class we will be focusing on these two functions. One is the function PRComp, and then we will talk about the package Factomine R, where you can use these functions to do principal component analysis. We can then also visualize, right, the results of the analysis, because visualization is a very important component to understand what actually happened after the analysis, and also if you want to look into the data structure or the kind of clusters that we have in the data. Okay, so let us now move on to the hands on part, and let us do the principal component analysis with our data set. So here is the R console, right. So we have loaded the data. If you remember last time, we have a cancer data set where we had 10,000 samples. The original data had 10,000 samples, and expression levels for 103 genes along with the labels of the samples, right. So these labels are different cancer types. From that we processed that sample and we chose only a small number of genes, so 20 genes and only 60 samples. Okay, so this is something we did just to make sure that the analysis runs on a short period of time, otherwise we will have to wait for a long time for the analysis to complete. But I will again encourage you to run this analysis on the full data set if possible, and see how the results come out. Okay, it will of course take a bit of time and you have to be patient. Okay, for now let us focus on these techniques and let us understand what is happening, right, with this analysis and the code. Okay, so here we are after the last class. So we have processed the data, we have made this new data set, right, where we now have only 20 genes and 60 samples, and we have removed the labels because we are not going to do principal component analysis on the labels. But the label is important to retain, because later on we want to see whether the cancer types, samples from different cancer types, they cluster separately. Okay. And this is a very common kind of analysis visualization that we do often with big data sets, right. So the first principal component, once we do, the first thing we want to look at is whether depending on specific sample types, whether the samples separate out. Okay, and that will mean, okay, there are features or genes or specific features that you are working with that could separate out these samples. So we can use those features for predictive modeling for machine learning. Okay, so let us now do the first principal component analysis using this PRComp function, as I just mentioned. Okay, and this is a built-in function in R, so we do not have to use any specific package. Okay, so here it is. So the data is actually this new data that we have just created, and what we are doing is we are scaling the data. Okay, I'll just very briefly show you what happens if you do the scaling, right. So this original data you can already see, right, these expression patterns. Scale data, so we can is actually simply kind of changing the transforming the data so that we kind of in the same scale, right, the same range. Okay, so we move the origin to 0. We are not so we are not doing this centering also, right. We can also do something called centering, right, where we actually changing the origin to 0. Here in scaling we are kind of maintaining the range, right, similar for all data sets. Okay. So, what we will do is we will do the principal component with the scale data, we will not center the data for now. Okay. So, what we are getting, let us see, is here, and what we will see is the principal components which are given here, as you can see, is PC1, okay, PC2 and so on. And what you get for, we have 20 principal components, right. So, we have 20 genes, so we have 20 principal components, and what you get is the value of each of these genes. Okay. All right. So, this is what we kind of have looked into, right. These are the principal components. Now, what we want is, so is the summary. So, let us do that. Okay. And you have this principal component PC1, PC2 and so on, 20 principal components. What you need to look at is this 0.5089 is this value, right, proportion of variance. This is a parameter that we are interested in, okay, PC1, PC2. So, what it means is proportion of variance is that 50% of the variance is explained by this principal component 1, okay, and then followed by 0.1769 means 18% of the variance is explained by PC2, and then you have PC3 which explains 6.5% variance and so on. So, as you can see that principal component 1 is explaining the most of the variation in the data, about 51% or 50 or 51% in this case. Okay. So, this is something that is important to look at, and then we also have the cumulative proportion. It is simply like how much total this PC1 plus PC2, how much of the total variation this PC1 plus PC2 combination explains, right. So, it is simply the cumulative, and the total is 1 here if you go to PC20. Okay. Now, what we also want to look at is something called PCA rotation, okay, and we have seen this already, right. This PCA rotation is actually the contribution of each of these 20 genes to each of these principal components. So, we have PC1, PC2 and so on, right. As you can see that weightage is different for different principal components, and it is not always necessary that all genes would have to contribute to all PCs. Okay. Here it's we are, because we are working with a relatively fewer number of features, so that is not an issue. Okay. So, we can also look at, right, the components in the first sorry, maybe, yeah. So, this is the first principal component, right. So, we are looking at the rotation first column. So, principal component, what is the weightage of each of these genes, and what we can also do is we can sort these, right, values and then in decreasing order. So, what it means is that these, so if we can sort these rotation values, right. So, these are actually the loadings, and we have sorted them according to their magnitude and decreasing order. So, the highest contribution will be the first and the least contribution will be the last, right. So, these ones, right, these genes, for example, NAP1L3, right, this contributes the most towards the PC1, right. So, what we also probably can do is something called absolute value, right, not just sign, but magnitude. So, if you look into the absolute value, because some of these genes also show negative contribution, right, and we can also look into the absolute values of this contribution. Right. So, what this means is that these genes, right, that are at the top, they contribute the most toward the fast species, right, and this is something that we will have to be kind of could be interesting, right, if we want to use these features for building predictive models. Okay. And then the something what we will do is we will plot now, right. When you say plot PCA, right, this is where the PCA results are stored, right, and as you can see, this kind of tells us, right, how much of these variances are there, right, and explained by this species. And the other thing that we can also do is something called biplot. Okay. So, what is biplot is simply this, right? Biplot means we have the data points here. So, biplot is simply looking at the expression or relationship between the different features that we have. Okay. And one of the things that we also need to remember, right? So, for PC1, as you will see, if it is not clear, so maybe you can zoom in a bit. So, for PC1, you will see some of these features, right? You have this negative direction, right? So, they are actually contributing to PC1, right? So, if you remember these absolute values, right? So, these absolute values of these loadings are there. So, this is actually telling us how important these features are for PC1. For positive contribution, we got these genes, right. So, NAP1L3, this is the gene that had the highest positive contribution towards PC1. So, you can see this here, right. So, the features or the genes that contribute to principal component 1, they will be parallel to PC1, okay. And the features that are orthogonal, right, they are perpendicular to PC1, they contribute the least, okay. So, you can see this, for example, ABI2, ATP6, V1G2, these are the genes that do not contribute that much to PC1. But they are parallel to PC2, right, close to parallel, or there is some if you take the component along PC2, they will contribute to PC2. Okay. So, this is something that we can now understand, right, what are these loadings and which genes contribute, which features contribute to which PCs. The other thing we can also look, if we have the label data as well, we can see which of these features are correlated with labels or which contribute to which are parallel in the same direction as the labels or not, right. So, this is something we'll leave for now, right, we'll look into that a bit later. Okay, so, this is something we can do very basic, but of course, there are a lot more things we can do in much nicer way, and we will use this package called library GG45. Okay. So, we are loading this package. So, again this will help us in visualization. So, the command that we are going to use is called autoplot. Okay. And here is the data autoplot. As you can see, PC1 explains 50.89% of the variation, PC2 explains 17.69% of the variation. And autoplot, what we are going to do now is we want to color the labels, okay, color the samples. So, these are, each point here is a sample, right, and we want to color the labels. The PC component, the PC1, PC2, these are combinations of the genes, right, the features in this case of the genes, right. So, these are in the PCs, whereas this each point here is a sample, okay. And what we are doing now is we are actually labeling this or coloring these points according to their labels, right, cancer type labels, and you can see this coloring here, right. ACC is red color, BLC is green, and LML is blue. So, what we see now is in this structure is that actually these data points separate out, right, the samples are actually separate out. So, blue points are actually closer to each other, green points are closer to each other, and red points are closer to each other, right, and they are kind of distinct positions in this plot. Okay. So, that is actually a good sign, which means each of these cancer types, they have the specific gene expression patterns which can be distinguished by these PCs that we have, right. So, and these PCs are simply combination of genes. So, if we look into the genes that actually that have highest loadings in PC1, they are likely to be very important features, right, for separating out these 3 different cancer types, and similarly combinations of PC2, right, that also help in separate out these different types of cancer samples. Okay. So, this PC1, PC2 now they are able to distinguish between different cancer types with the help of only 20 genes. Okay. So, this is something that is very interesting. We can actually also make this, right, extend this analysis to actually do a 3D plot where we also include PC3 and see where that actually leads to better analysis or not, better visualization or not. Sometimes what will happen in 2D plot, some of the sample types will be overlapping with each other. Okay. And in those cases, we would like to use the 3-dimensional plot, which might give us better view. The other thing we also need to remember, sometimes the first principal components may not be able to give this separation, right. Here we got this separation for different cancer types very easily with PC1, PC2. Sometimes maybe other PCs, like PC4, PC5, they can separate out samples types depending on the data that you are working with much better than PC1, PC2. So, that is something that is worth looking at, and which we will do in the second example, right, where we will actually look into different sets of PCs. Okay, so this is something that's actually good for this moment, right. We have the blue, green and red samples separated out. So, this is very good. Now, we can also kind of make this a bit kind of look into, we can kind of make this cluster, kind of highlight these clusters, right, with this kind of plots, right, where we can have a frame kind of thing, right. So, we will say frame equals to true. Now, we can actually look into these different frames, right, kind of they are kind of containing the samples. And the other thing we can also do is we can put the loadings here, right, like before, right. In the peer comp analysis we actually did that, and here also we can, on top of these points, we can also put these loadings, PC loadings. And as I mentioned, right, we interpret it in the same way, that genes that are parallel to PC1 or have some kind of projection on parallel to or projection on PC1, right. So, those will have some contribution to PC1, right, whereas the ones that are perpendicular to PC1, that are like they will have only very little contribution to PC1, right. So, this is something that we can now interpret and understand. Okay. All right. So, this is the first package that we have used. The second one that we are going to use is called Factomine R. So, this is a specific package, right, where we also have a PCA function, and this is what we are going to use now. And let us load this. Okay. So, again we have to be careful about the spelling and the capital and small letters. Okay. So, this is done. The other package that we will also use is called library factor extra. So, this is a package that we will use for visualization of the PCA analysis. Okay. So, the command that we need to use with this package is called simply PCA, right, in capitals. So, we have to use this command. We will do this with data new, which column names not equal to label. So, we are not going to do PCA with the label column. So, that data we are not going to use, and we do not want any graph, because whatever plot we are going to do, we will do them later on. Okay. And let us look at this, what we have. Okay. So, here are some of the results, right, and here it mentions, right, the analysis was performed on 16 individuals described by 20 variables. Okay. And the results are available in the following objects, right, and here are saying eigenvalues, results for the variables, coordinate for the variables, etc. Okay. And there is a lot of things in here. So, what we are going to do is, let us look into some of this, right, and that is the first part we will look into, right. So, we are looking into the results for the variables, and what we have here for variables, we have coordinates, we have contribution and correlation, etc. So, what we are going to look at is the contribution part. Okay. And again, the contribution is kind of summarized here for dimension 1, dimension 2, dimension 3 and so on, and you can again take this and kind of sort these values, right. We are taking the first column, and what we are going to do is decreasing equals to true. Okay. And here you see these genes again, right, this RNF169 that has the highest contribution, followed by LAT S1, ASXL2 and so on. Right. Again, we have only 20 genes in our analysis, but if we include more genes, of course, the results will change, and maybe there are other genes that will contribute the most. Okay. And this is something you can try out, right. You can take more genes, right, 30 genes, 40 genes, 50, 100, all 100 that we have. So, all those analysis you can try out on the same set of sample, and you will see how the clustering might change, and how these loadings, PC loadings might change. Okay. So, here just to be clear, right, we are looking into the dimension 1, this is the PC1, and here we are looking into the contribution of these genes towards PC1. Okay, and once we sort them, we can find out the most important feature that contributes the most. Okay, so this is done. Now what we can also do is, so this is something we have already done, right? So we have looked into the most important feature by sorting. So this is what is kind of written here in these commands, which we have done already. What I will do next is something called a script plot. Okay, and script plot is simply this, is where we have this list of PC components along x-axis, and y-axis is the percentage of variance explained. Okay, so that's what we get in script plot. In here, we are going to use this function mvis underscore I. So this is actually from factor extra package for visualization and the results of the PCA2. Okay. So here you get this, right, where we have the dimensions 1, 2, 3, 4. So this is actually the principal components 1, 2, 3, 4, and y-axis is percentage of explained variance. So as we have seen, right, the PC1 actually explains 50% of the variation, followed by second PC, third PC, and so on. Okay. Now what we can do next is this, right. So we can, there are other functions in there, so you can look into this package, right. There are other functions in there, and again we are plotting this again very similar, right, dimension 1 versus dimension 2. Again in the bracket you have 50.9% of the variance explained, here 17.7% variance explained, and these are the data points of the samples, right. So we have 60 samples, these are present on this plot. Now very similarly, right, we can also color this, right. Okay, and the command that we use is called happy life. Okay, so this is the command that we have to use here with this factor extra package, right. Happy life is the command, and this is simply this. So every PCA individual, right, the results of the PCA, don't put the labels. So if you put the labels, of course you will have the sample labels along the plot, which we don't really need. And then when you use this command as factor data new labels, right, so it will create colors, different colors for different samples, right. So different cancer types that we have. Okay, now the moment we use this, we get these different colors, as you can see, right. We have again have red, blue, green clusters, and this is something that we have seen before. So this is actually good for our analysis, because then we know different cancer types, they can be separated out using only these 20 genes, right. So and some combination of these 20 genes. We can make this a bit more complex, right, and then what we have is, okay, so we kind of building on this, and then like there we can also have add ellipses, right. So earlier we used this add frame, right, we added frame. Here in the GG45 package, here we can use this add ellipses, and ellipse level 0.95. So 95% of the data points should fall within the ellipse. Okay, so again this is something we can do, right, and this is kind of like summarizing, right, where all these samples are present, and 95% of the data points would be within the ellipse. What you also notice, right, these some of these samples, red samples, they also fall within the green ellipse, as well as some of the blue samples, right. So this is something is because of the structure of the data. Anyway, this is something that we kind of can do. We can further do something more, right. You can say, let's see, right, here we are now adding these loadings, right, as before, like in GG45 we also we can add these loadings, and again from this we can kind of understand which genes kind of contribute to PC1, which genes or which features contribute to PC2, and so on, and also the relationship between these genes, right. So we kind of see this, there is kind of these genes are kind of in opposite direction, right. So what happens is that in some samples maybe this gene is highly expressed, and in other samples it's in some samples where so this opposing relationship, what it means is that if this gene is showing certain pattern, right, and compared to the other one, it has an opposing pattern, right. So if it is, just as an example, if gene 1 here is highly expressed, and this gene too is lowly expressed, right. So, that kind of opposing relationship, whereas in the other set of samples this gene is highly expressed, the other gene is lowly expressed, right. So, that is something again based on the direction of these features we can kind of also understand the relationship between the features themselves, okay, as well as their contribution to the dimensions or to the principal components, okay. So, we can kind of do a bit more here, right. So, here again it is the, you can see the contributions, right. We have the dimension 1, right, and what are the contributions of dimensions of these variables towards dimension 1, okay. Let me see, yeah. So, color by contributions, right, color dot var by contribution, color them, and gradient colors, right. So, these are the gradient that we are using. So, this red orangish color means they have the highest contribution, this yellowish means there is some contribution, right, and green means almost no contribution, right, they are perpendicular to this dimension 1. So, there is almost no contribution, okay. So, this is contribution towards the PC. We can also do here something called a PCA byte plot. So, this is the command is, we have used the byte plot command, we have used this var command, right, and again these commands are all described in the manual, okay. So, we now know how to look into the manual and what these commands actually do, right. So, what we have done here is we are again kind of looking into the byte plot. So, this is what a byte plot is, right, where we actually looking into the variables and their kind of relationship with the principal components of the dimensions as well as the relationship between them, okay. So, this is what we refer to as byte plot, okay. So, here is the results, then repel equals to true, otherwise there will be kind of overlap between the names, etc. Color variable is red and color individual point is blue. So, these are the data points, these are shown in blue color and these are the kind of number which data point is which number, etc. And the variables here, the features or the variables are shown in this red color, right, with the arrows, okay. So, this is something what we call as byte plot. All right. So, I think we have done the first part where we have looked into the principal component analysis using the PRComp package, as well as we have done some visualization with GG45. Following that, we have done this PCA analysis with the FactorMineR package, and we also use the FactorExtra package for visualization. And we have also learned about some of the how to interpret these plots, right, the visualizations that we have made. Okay, so let us go back to the presentation and let us summarize what we have done so far, right. So, we have introduced a specific data set, a cancer gene expression data set, where we have selected as only a small subsample of that, and we have done the first PCA analysis in R. We have used two functions. One is of course the R function PRComp, and we have looked into the PC loadings. We have done the data visualization, script plot and byte plot. We have also used the FactorExtra R package, FactorExtra R package and FactorMineR package to actually do the principal component analysis, and again we have visualized using the FactorExtra package. And what we have learned is the script plot and what it means, and we have also looked into a byte plot, which kind of tells us which features are important for differentiating between different samples and how they actually load the principal components. With that, we will discuss the other tools in the next class. Thank you.